So here's an outline of what we're going to be talking about over the next hour and 40 minutes, just a really basic outline. And so we're going to go back and forth between the two of us. And I won't go into great detail here because you'll see it flash every time we transition between one of these 9 points. After we introduce ourselves, I'll start things off by giving a pretty broad introduction of what bioaerosols are from both the outdoor environmental perspective and the indoor clinical infectious disease perspective, and that's where we'll transition more into what Josh will be saying. And then I'll also come back and talk about some real-time techniques and then some field work, and then we'll go back to him. So we'll go back and forth a little bit, and we'll flash this each time. Next, please.
Yeah, so this is me, Dr. Josh Santarpia. I currently at the University of Nebraska Medical Center in the Pathology and Microbiology Department. I also have affiliations at our University Affiliated Research Center, the National Strategic Research Institute. I manage our program in biodefense and health security, at least for now. And I'm also a adjunct faculty at our Lincoln campus. So I get around a lot in the state of Nebraska for sure. Most of my work overall has been around the biodetection types of applications or detecting disease-causing microorganisms. Most recently, that's been related to SARS-CoV-2, particularly in the clinical environment. So, Alex?
Yeah, and as I said, I'm Dr. Alex Huffman. I'm at the University of Denver, so I'm an associate professor in the Department of Chemistry and Biochemistry. So I have my degrees in both undergraduate and graduate degrees in chemistry, a PhD from CU in Boulder in atmospheric and analytical chemistry. And the work that I do in my lab, and this is a picture, a little bit outdated group picture, but we work on all things aerosols and especially bioaerosols from the outdoor perspective. We've done quite a bit with instrument development and characterization and applying those instruments to outdoor and indoor field applications, looking at pollen and fungal spores and all bacteria and all sorts of bioaerosols we'll talk about in a few minutes. I've spent quite a bit of time working on fluorescence detection, which we'll highlight as well. Over the last year and a half, as an aerosol scientist, I've put a lot of energy also into the COVID pandemic from a different angle from what Josh was talking about. I've spent a lot of time advocating and working with school districts and various different places to try to educate on the airborne nature of the spread. And so actually on the right side here, there's a QR code for some of the educational information I keep updated on a website that shares media articles and scientific articles, various different infographics, things like this, just to educate the media and the public, and as well as keep resources for scientists. So, and also fairly active on Twitter as well. This handle on the left side, Huffman Love to you if you want to look at anything there. Okay, Josh. Okay, so that is the introductions, and so from there we're going to go into a broad introduction of bioaerosols for a little while. So go ahead, Josh. So normally I would have animated some of these slides, I should say, but because I am not in control of my own clicking destiny here, I decided to make it easier on Josh by taking out those animations. So some of the slides are a little bit busier than I might normally have, but the one broad way of thinking about bioaerosols is listed here as in a definition that was written in 2012 by Viviane Desprez in this Telespe article, and I'll just read. The term primary biological aerosol particle, PBAP, is defined to describe solid airborne particle derived from biological organisms, including microorganisms and fragments of biological material such as plant debris and animal dander. The definition of PBAP can thus include all sorts of intact fragmented biological cells, dispersal units, or tissues. And so when I say, I may go back and forth between certain terms, and it's very possible that Josh will use some other terms that are related to this, but there are some soft edges to the definitions here. But the idea is anytime you have direct biological material in an aerosol that's floating around in the air is usually wrapped up into the term bioaerosol, or in this case, specifically termed primary biological aerosol particle. It's depending on your research perspective or your interest perspective, there's a term biogenic aerosol and biological aerosol. Biogenic aerosol simply means it comes from a biological source, but it doesn't necessarily have to be biological in nature itself. Biogenic could mean gases that come off of trees that then photochemical react and then condense into aerosols. So that we would usually consider to be biological, but biogenic. And in the respiratory environment, the indoor environment, there are also fuzzy edges to this, and different communities may call these particles slightly differently. And I'm not going to get into all the debate about droplet versus aerosol and all of that process that has been crazy over this last year. But the idea being any aerosol from this perspective of this presentation that has direct biological material in it, I'm just going to call bioaerosol. I'll let Josh say that if he changes it a little bit. And so on the lower right, you'll see simply a size continuum where over however many 9 logs, we have particles that start as from the biological perspective, you have individual proteins that can be tens of nanometers in size. Viruses, as we've heard quite a bit about, can be tens to hundreds of nanometers in size, but viruses don't float around by themselves, as we've heard about. They're encased in bigger droplets usually. So bacteria, a few microns, spores, a few microns to tens of microns, all the way to pollen, which can be tens to hundreds of microns. And so we have a really wide range of even within the biological particle spectrum. Okay, next. So bioaerosol research itself is intensely multidisciplinary, as you have even heard discussed in terms of the respiratory side of this and indoor environments. And so this is just a snapshot of a paper we put together a few years ago. And I highlighted here, it says the key challenges in the study of bioaerosol include inherent multidisciplinarity of the work, often involving not only aerosol science, microbiology, atmospheric science, physics, chemistry, and engineering, but knowledge of building science, ventilation, sampling, human dynamics, medicine, public health, infectious disease, botany, and so on. And so you can see how this can be complex when applied into a research environment, because it requires the collaboration between people that often don't sit in the same room or talk to each other. And so we've got to get on the same page with definitions and thought processes, things like this. Okay, next. And as we talk about both bioaerosols and then the sampling and the technique aspects of this, I just wanted to highlight this. This is a special issue in Aerosol Science and Technology that we put together a couple of years ago. So Josh and I are both on a couple of these papers. It actually came out of a discussion at the AAAR conference a few years ago. And so if you're interested in this, it was published in 2020, volume 54. And so there's 9 really nice review articles there that I won't go into detail on talking about here, but it touches into all aspects of the technical and the research side of bioaerosols. Okay. And so I'm going to talk a bit over the next few slides about where bioaerosols come from. And this means I'm going to talk a little bit about the emission mechanisms, which then helps us think about what bioaerosols are in each of these different environments. I'm not going to say them all here, but this is a nice book that was also published relatively recently, and it was edited by Anne-Marie Delort and Pierre Amato. And it goes through, especially from the outdoor environmental perspective, but also has some aspects of the indoor human emission side of things as well. Next. So the first that we'll talk about in terms of the type of bioaerosols and also one emission mechanism is how spores can be actively emitted. And so let me just pause here to say that I'm going to highlight several different emission mechanisms that then also talk about several different specific types of bioaerosols. This is not meant to be a comprehensive survey of this. I'm going to highlight some important ones, but there are others as well, especially if you dig in some of the resources that I just said. But these are some of the important types of biological particles that have certain relevance within aspects of our community. And so I'm going to talk about emission mechanisms. Then I will talk about a brief overview of why we might care. So fungal spores can be emitted actively when one of a couple different mechanisms take place. On the left side, you see an osmotic pressure cannon. In the middle, you see this Buller's drop. Buller's drop, they both have, well, the Buller's drop in the middle, the idea is you have the spore that grows on the fungus, and where this blue dot or blue arrow is pointing, it secretes a little bit of hygroscopic material, some things like mannitol, arabitol, potassium, and some other salts and other materials. And these are hygroscopic, so in a relatively humid environment, water condenses onto that little dot. And so as the sequence proceeds to the right, you can see Buller's drop form, and the water droplet forms and gets bigger and bigger until this the surface it may have the rehearsed timing on it. As it goes to the right, the water droplet gets so big that the geometry is such that it can't stay there anymore, and it kicks the spore off, and it fires off at fast velocity. And so I put this quote down here. It says, the subject to this is just sort of the cool factor of bioaerosol emissions here, subject to an acceleration of 25,000 G, which is about 10,000 times the acceleration experienced by astronauts during the launch of the space shuttle. So that's what these little spores do when they fire off. And so they also grab some of this mannitol and potassium, other things. And so we can get some clues in the atmosphere when we see these things. So these are fungal spores of different types. Okay, next. So I just mentioned that some of the potassium is attached to these fungal spores. It turns out that the potassium that gets launched into the atmosphere as a result of this and other similar processes can actually become seeds for other the deposition and condensation of other organic materials. I'm not going to get into this relatively complex slide by Christopher Proker from Science in 2012, but the idea is in the Amazon, you have enough potassium from fungal spores that it doesn't, that the organic material can condense onto some of these small seed particles, and it goes into the whole processing of SOx, secondary organic aerosol, and primary organic aerosol. And this heavily influences the whole cycling of aerosol in the Amazon. And this doesn't take place in other forests like Huttula in Finland as much, because you don't have as much of this potassium put out by fungal spores. So it can really impact the whole geochemical cycle. Okay, next. Another really cool thing from my perspective is the bursting of pollen. In fact, there's a little GIF here that thankfully is working. It will recycle after a second. And so I pointed to originally where the pollen starts, but if you put certain types of pollen in water, or if there's enough relative humidity around, they can swell up, and they eventually get so big, they're like a water balloon. You put too much water in it and they explode, they get a little pop, and they fire out the contents into the air. So a pollen itself, as you can see on the bottom, this one, for example, is something like 25 microns, but then when it pops, the contents can be much smaller than a micron in size, and these cytoplasmic materials can go all over the place, and the allergenic material from inside can be important. The small particles inside may have CCN activity, as people have looked into. And so there's all sorts of things that can happen from this once the pollen explodes. Go on, Josh. Here's another mechanism of bioaerosols getting into the air and outdoor environment. And so this is a GIF that is from Lydia Bariba's group at MIT, showing bubble bursting. And so this is the same mechanism by which particles enter the atmosphere from it doesn't have to be a bioaerosol to get into the atmosphere this way. But if you have biological particles on the surface of a water body, whether it be the ocean or a lake, and you have bubbles form, as shown on this left side, we have waves crashing, makes bubbles, and then when the bubble forms, the films break up, and now whatever was on the film is now in the air. There's various different processes. You can get into microphysics of what's taking place here, but bubbles that are entrained allow biological particles to be emitted into the atmosphere from water surfaces. Okay, go ahead. Same idea when you have biological material interacting with water, in this case, rain splash. You have rain coming down. If you have bacteria, let's say, on this, or some kind of a plant pathogen on the surface of a plant or any other surface or the soil, the rain comes down, splashes, and then it launches this material up into the air. And so again, some of these pictures are from Lydia Bariba's group at MIT, and there's some really cool videos that she makes available here as well if you want to go find those. And the idea is that this is one mechanism by which you can get plant pathogens from one plant to another plant, even though the bacteria or the spore itself is not walking over there directly, but you have rain splash and then it gets up into the air and then it can descend into the nearby plant or the next crop field or something like this. So this is an important way that material can get into the atmosphere, also not just from a pathogenic perspective, but just simply in any natural environment. Okay, go ahead. So that was a really whirlwind tour of some of the mechanisms, and I just wanted to come back from the outdoor environmental perspective. The global PBAP emissions are significant. So this lists various different types of fungal spores, as we've talked about a little bit, from bacteria and on down. And so you can see on the left side, the emissions are in the teragrams per year, anywhere from less than a teragram. Pollen can be tens of teragrams per year, and fungal spores even up to hundreds of teragrams per year. And so in any given environment, this paper by Janine Fröhlich-Nowicki put together some estimates on the number concentration that you would expect to see in any given environment, anywhere from bacteria, 10,000 bacteria per cubic meter, or fungal spores on the order of 10 to the third, and then down. And then in terms of mass concentration, these are obviously estimates, and there's a huge range depending on where you are, but somewhere on the order of micrograms per cubic meter for most of these individually, either in the tenths of micrograms or individual micrograms, unless you're in some unique environment. So this is a significant fraction of overall organic material in the atmosphere in a lot of places. Okay, go ahead. So that's our first transition out of the introduction to the emission mechanisms of outdoor bioaerosols. And so I'm going to say a few things about some of the reasons we care and the why behind it. Again, focused mostly on the outdoor, and then I'll transition into Josh's perspective. Okay, thanks. So this is a one figure, not perfect, but one way of saying how the nature of bioaerosol research is interdisciplinary. So in the center, we have bioaerosols, and these different dots are meant to represent the different communities of people, of researchers that think about bioaerosols. So at the top, from the perspective of biodiversity and biogeography, going around clockwise, agriculture, the allergenicity of particles, human pathogens, both from an outdoor perspective, and especially an indoor perspective, to atmospheric chemistry, to cloud physics and hydrological cycles, and I won't read the sub-thoughts there. And you can imagine that each of these different communities have very different interests, motivations, drivers, funding sources, techniques, definitions, conferences. All of these things make it relatively complicated to merge these things into one continuous thought process and set of coherent and forward steps, which is why conferences like AAAR can be useful to bring people together, at least from one angle, to think about these things. Okay, next. So one of the reasons we care about bioaerosols from an outdoor perspective is their interaction with cloud processes. And so their ability to nucleate ice is one reason we may care. And so depending on how much you know about ice formation, liquid water doesn't freeze until less than about -38 degrees C unless you put something inside it. Depending on what you put inside it, it can freeze more efficiently. And so if you put certain things inside mineral dust and others, it doesn't freeze as efficiently, but certain kinds of biological particles can actually nucleate ice. at much warmer temperatures. Instead of -15, it can be all the way up to -2, let's say. Okay, next. And so you can go ahead, Josh. One reason, one source for this is for example, a bacteria called Pseudomonas syringae, although there are many others. I just chose one here. It turns out it has proteins on its surface that mimic the crystalline shape of ice, which is really cool. And so it basically tricks the water into freezing. And so this uses the bacteria uses it as it acts as a plant pathogen to crack open the plant so it can get inside and eat, basically. We also use this industrially for snowmaking applications. But more importantly, in the atmosphere, it helps. It's unknown yet to what extent this happens on a global scale or a regional scale to actually affect ice formation and precipitation, but that's the research process of this, to think that it may actually contribute significantly. Okay, next. This is just a quick plot on the x-axis to show the temperature of freezing, on the y-axis to show the number concentration, and you see this big chunk of stuff on the middle at about -30 degrees C of desert dusts, mineral dusts, soot, all sorts of other stuff. And then there's this on the right side. There are certain bacteria, these green circles that nucleate ice at quite a bit warmer temperatures. And so that's what we're talking about. Okay, next. And then the question is, do they actually modify precipitation in any way? And that's a big question. And one question at the bottom I posed is, could land use changes actually affect precipitation and back and forth? So there's certain kinds of bacteria, biological particles on a plant, and we chop on a kind of certain type of tree in a forest. We chop down the forest, those bacteria aren't there anymore. They're no longer going up to the atmosphere. Have we unknowingly changed the precipitation patterns downwind? And that's an important question for all sorts of people. Okay, next. And the biological particles themselves go up into the atmosphere, but don't stay as they were emitted. And this is just a one slide example of the idea that biological particles, as shown in the middle, can get emitted, but then they go through all sorts of chemical reactions. The next slide, I'll talk about physical changes. Here, I'm talking about chemical changes, where they can, for example, react with ozone or NO2 or various different oxidants and reactive radicals in the atmosphere, and then undergo changes that change their chemical properties, that then also change the way they interact with the clouds by changing their hygroscopicity. They can even metabolize in the cloud. There's various different ways that their chemistry, their active chemistry, can change the situation. But they can also, it's also been hypothesized that once the proteins have been modified, they can be more allergenic, and it changes the immune response. So there are a whole field of chemical effects of biological aerosols that we care about as well. Okay, next. And then I said that there are physical processing that can take place as well. And so this is one example of that, where I already mentioned the pollen can rupture, but if you take pollen, pull it in a strong updraft, let's say in a thunderstorm, now you have a lot of water and you have pollen that has been sucked into that cloud. And so the pollen can be in a really wet environment and then rupture. And then those ruptured particles, if they're allergenic already, now you take them, and instead of being 100 microns in size, which then as you breathe them in lands in your nose and the back of your throat, if they are now submicron in size, they may have allergenic material. Now you can breathe them deeply into your lungs, and so now it's not a nose allergy, it can actually cause severe respiratory issues. And so the term thunderstorm asthma is important, especially in areas of Australia that see these effects because of the biogeography and the meteorology in certain places. And so one example in the lower right, even from the New York Times, where thunderstorm asthma killed 8 people in one specific storm episode. And so it can be a really important issue. And this is an example of the type of physical processing that can take place. Okay, next. And so then this gets us ready to, I have just a couple quick slides here as we start to transition into Josh's area, but there are a variety of reasons that we would care about bioaerosols in an indoor environment. I didn't actually put a specific slide on mold spores and the damage that that can do, and then the human health effect that mold spores inside can have. We've talked a lot over this last year in the whole world about respiratory viruses. And so this is a one-slide summary of the idea that you can breathe out particles that have various different emission mechanisms that are actually mimicking some of the things that we just talked about. In the lower left, it shows an example of wave breaking that can produce particles just like we did on the ocean, but a mimic of this effect can actually take place in part of your respiratory system to then produce particles. And so there are various different physical processes that then allow particles to come out of your respiratory system that now have proteins in them, that they may have bacteria or viruses in particular, as we talked about this last year. And so that can be important. Next slide. And this is a really complicated slide that I won't get into, just to say that on the x-axis is a particle size distribution from 0.01 microns up to 1,000 microns. And so in the middle sections, it says tuberculosis and influenza, SARS-CoV-2, and different diseases have different, it's thought to be different size distributions of where the viruses come out, the peak size at which viruses come out based on where they exist primarily in your respiratory system. And so that can be modeled and thought through in ways that Josh will get into even more. And I have one more slide, Josh, that we can just skip through just as we transition into your slide. Just to say that aerosols are important both in the rest of the room, but also in the near field as well. So whether you have a big puff that gets in your face or whether it gets mixed into the room, they can both be important. Depending on the size of that, it changes how we think about it and also how we sample it. And so I think I'll just, for the sake of time, kind of skip through that and then give you the mic.
Okay. Yeah, so I'm going to start now, and I've got a couple of simple points I want to make before we start getting into more of the meat of the rest of the tutorial, which is going to be sample collection techniques and real-time data measurement. And I think that, while I'll be largely covering sort of sampling and analysis techniques for bioaerosols, and Alex will largely be covering real-time measurement of bioaerosols, we cover them from sort of the perspective of infectious disease monitoring and the collection piece and environmental experimentation in terms of the real-time piece. But in principle, both types of techniques are used in both fields. I do feel like that they're a little more common. The outdoor environmental type measurements are more commonly used. Real-time methods rely heavily on real-time methods, whereas I think the indoor infectious disease kinds of aerosol methods rely more heavily on the collection analysis piece. I wanted to put this up sort of as a follow-on to Alex's mask thing and look at the evolution of PPE, vice our understanding of how respiratory or other diseases work. We have the plague doctor with the flowers stuffed in his mask to prevent the bad air or the miasma. We have the early sort of medical nursing practice, and I think this is more when we think about the things that we can see. Like it's always the observable, right? When we didn't know anything, it's miasma. We can start to observe these sort of. I have this really nasty picture of this guy sneezing into a cloth, but the idea there is these aren't like heavily preventative in terms of aerosols, but they would likely prevent most of the contact with droplets. And then as we evolve our understanding and begin to understand more about the roles of aerosols and the transmission of respiratory diseases, our PPE goes forward, and we start looking at things that are more like our N95 or the respirators, PAPRs, face shields, as we start to think about the actual mechanisms of disease transmission. And so I want to come back to a little bit of Alex's slides before, where we just looked at the sizes of sort of the root, the actual microorganisms that are in biological aerosols. And I'll say this about infectious bioaerosols. I think it's maybe less true in the environment. Alex showed several examples of fungal spores and other things being released, and I think those are more in their natural state. Infectious bioaerosols are rarely, if ever, seen as their sort of naked microorganisms, right? There's always some sort of matrix. If you think about coughs or even just exhaled breath, there's a virus in an existing respiratory droplet that probably has an existing size, and I have some data from Nebraska in 2009, looking at the modes of respired particles, and it's basically viruses and smaller microorganisms fit inside those respiratory droplets. And in fact, the composition and the behavior of infectious bioaerosols is often dominated by the matrix that they're in, whether it's that deep lung fluid or saliva or something else. A lot of times that behavior is really controlled by the matrix rather than the organism. So I think it's an important concept to think about when you're thinking about both, the size, how important is it that SARS-CoV-2 is, 70 to 150 nanometers? Well, it's important in that it can fit into the smallest of our respiratory emissions. And furthermore, those come from our deep lung where the infection happens, thus creating the idea that very small particles can be involved in the transmission of respiratory diseases, especially ones of pneumonia, and how that plays out in terms of what we think about the droplet and aerosol dichotomy. And Alex stayed away from it. I'm just going to mention it as we move forward. I don't think there is a dichotomy. I think it's all a spectrum, right? I prefer to look at aerosol transmission rather than sort of an airborne or droplet transmission, because it's a spectrum of emissions from the human, and which may or which for in any individual disease, as Alex pointed out a slide or two ago, is probably controlled by where the infection is and what size of particles are emitted from that part of the body. And so I think a re-examination of that scientifically is happening right now. I think it's been, I think a lot of the aerosol community has been heavily involved in that. I think it's a good thing. I think it's long overdue. So the other thing I want to talk about is the aerobiological pathway. This comes up a lot in infectious aerosol research, and in terms of how we break it down. Alex mentioned something before. We think about sources, whether that's coughs or sneezes or natural sources of infectious particles. There's a transport factor, so it has to get from the emission point to the person that's going to be infected. And many environmental factors, in fact, some of the same ones that are studied as a part of trying to understand atmospheric chemistry or impact on the cloud cycle, also impact the disease or the disease-causing microorganism on its way to the human, which is the health effects side, or the third portion of this aerobiological pathway, where the organism, and if it's a stable one, survives the transport and has some impact on the human after inhalation. And whether that's allergy or whether that's disease depends largely on what the source was. Whether or not a disease-causing microorganism survives long enough to reach the human and cause disease depends on its stability and how well it survives in the atmosphere in the matrix that it was emitted in. And all of these little factors play a huge role in how this entire process works. And so I'm going to talk about a lot of these sample collection and offline analysis. A lot of times we need these pieces to sort of pick apart. We got to collect particles at different times and in different situations to pick apart this, the pieces of this aerobiological pathway to understand the sources and the infectious disease portion. And so you'll recognize, if you've been around aerosol science for a little while, all of these particular techniques, impactors, maybe not impingers so much. Impingers might be somewhat unique to the bioaerosol world, but certainly not filters or cyclone collectors of any kind. These are all common techniques to all of aerosol science, collecting of all aerosol types of aerosol particles. There are some unique features that each of them have in terms of trying to preserve microorganism viability or not. But many times what you'll find is that a lot of bioaerosol collection technology is constructed around trying to preserve the integrity of the microorganism as you collect it. So, and there's a lot of debate and a lot of research around this process. I start with filtration. It's the most straightforward and simple means of bioaerosol collection. It is probably, despite sometimes my wish to do something better, it is probably the thing I use the most. Most of your losses come from extraction. So we'll talk a little bit about ways to get around that. And there's a wide range of downstream analysis techniques. But and I highlight a little caveat there. There's some limitations for fragile organisms, especially if you're trying to preserve integrity. And there's a wide range of filter media for different kinds of applications. There's metal filters and membranes and gels, glass fibers, and we'll talk a little bit about all of those. So Teflon and glass fiber filters, they're both suitable for lots of downstream analysis. Neither is generally suitable for culture, and I didn't put the word generally in that in this bullet, but I think there's a lot of microorganisms now, spores in particular, and even some viruses we'll see a little later on that don't seem to have as much of a problem being collected very aggressively by dry filter, as long as you don't overly desiccate them, I think, with long sampling times. The nice thing is that these filters are very glass fiber, Teflon filters, it's all autoclavable. The holders are autoclavable. You can chemically treat them to remove DNA and RNA, so they're really good for downstream molecular analysis. So you can have a very clean collection substrate and be able to analyze downstream very confidently for nucleic acids and proteins, and which is a little less so when you start to get into some of your liquid reagents where you might not have as much control. Teflon particles are a little better for something if you want to do microscopy or Teflon filters, sorry, because the particles will stay on the surface, whereas like a glass fiber filter, they're going to be embedded in the filter fibers. Teflon filters and actually glass fiber filters, if they're unbound, glass fibers will partially dissolve in water, which is nice from a recovery standpoint of the material. Gel filters are becoming, and especially even by me, common for and useful for viable bioaerosol collection. It's actually surprising how well they work. Some exceptions might actually be in some vegetative bacteria are a little too sensitive. But the gel filter itself is designed to reduce stress on microorganisms during collection. There's a little bit of water absorbed to the gelatin in the filter, and so it helps maintain some friendly environment for the organism upon collection. Of course, if you sample too long, you'll actually desiccate the filter, and then you basically not only will the filter crack and stop functioning correctly, but you'll also sort of lose the water in the filter that you were hoping was going to preserve your microorganism. Which is why, generally speaking, sampling is 30 minutes or less. You sample very gently, and in a not super dry environment, you can actually extend that. I've had success with these, out to several hours in certain situations. On the converse side of that, if it's very, very humid, they're not very good for outdoor applications in high humidity. I've had very poor luck with gelatin filters working with them outside in a place like Houston in the summertime, where humidity might be near condensing levels, and you can actually get the filter to melt onto the filter holder, which makes it all but useless in the recovery process. The nice thing is it's soluble in both water and buffer. So depending on, you can use either depending on what your, so your downstream analysis is. Impaction. So you can use a standard impactor for collecting bioaerosols. Again, probably if you're not trying to help the substrate a little bit, you're probably not going to wind up with a lot of viable preservation. But again, if you're primarily looking at downstream molecular analysis, it's not that important. But you can also impact on auger surfaces, like a slick auger sampler, which is a very kind of old technique. They have viable cascade impactors, which may have auger or water or buffer in the different stages. I've also had a lot of luck with some of the smaller samplers using gelatin filters rather than the PTFE filters they recommend as the collection substrate. You can actually do that pretty well and still recover some viable microorganisms in that way as well. So there are a lot of options when it comes to the adaptation of impaction type collectors for bioaerosol collection. Cascade impactors, again, just sort of dig this in. The Ciotis is one that's fairly common right now, as are the NGI, next generation impactor. These are all things that are being used pretty routinely in bioaerosol study right now. Another technique that's become fairly common is a dry cyclone. The NIOSH BC251 has been used by many people in the study of both SARS-CoV-2 and influenza to collect microorganisms. It was largely designed for the collection of for downstream molecular analysis, not assuming that culturability or viability might be preserved, although I think in many cases it is. We've repurposed. So again, a lot of times if I'm trying to preserve viability in cases like this, I think there have been people who've experimented with some amount of liquid in the cyclone. That's had some mixed results, but also a gelatin filter in the filter holder portion can help preserve viability in that stage. We'll talk a little bit about in sort of very toward the end how a lot of viruses aren't as, it might not actually be as unstable in collection as some of the larger vegetative bacteria. So another way that people use cyclones is a wetted wall cyclone, where you constantly feed a slow liquid drip. There are lots of examples of these wetted wall cyclones out there. And essentially you get like a slow liquid drip in there so that the aerosol is actually collected on the liquid surface and then recovered out later on. Some of the downsides of a wetted wall cyclone or any kind of liquid collector that we'll talk about is that you're, especially with a cyclone, because typically you're using very high flow rates in these, if you're using any kind of buffer, you'll essentially concentrate salt in the collection fluid if you collect for too long, and you can actually osmotically stress your organism in collection. So you don't want to run these too long, again, for the same reason that you don't want to run a lot of liquid collectors too long because of evaporation, because you can wind up stressing your organism. And so again, yeah, so back to impingers. Again, these are both like aerosol to hydrosol type collectors. Most impingers were designed to mimic a human resting breathing rate. And so they essentially were designed originally to sort of mimic both, collection of aerosol by people to look at exposure, but they're oftentimes used, and in fact, some of the more like the SKC biosampler that I've shown here are essentially designed to collect a very large fraction of the particles, not necessarily representing the human, what gets lost in the human respiratory tract, but really try to be more, higher efficiency. And again, so impingers are designed generally to work at sort of human breathing rates. Wetted wall cyclones, as I said previously, are designed to work at very high flow rates. Both techniques can preserve viability. However, as I said, both techniques are susceptible to evaporation during collection. And so collection times or collection fluid have to be really carefully managed so that you don't stress your organism by artificially increasing the osmotic stress by evaporation and concentration of salt in the collection fluid. Electrostatic precipitation is something I have to cover these days. I think last night I made some changes to this slide because this technology is becoming really widespread. I think the first time I did a presentation covering electrostatic precipitation, it was still sort of, it wasn't as pervasive as it's becoming in bioaerosol collection. It's been a common technology for particle collection or removal in other parts of the field. For bioaerosols, it has a huge advantage of being the collection forces are generally small by comparison to even some of the gentler techniques we've talked about, and so you can, and it's controllable, so you can actually make lower collection forces by changing the basically the dimensions of your collector. Manelis and others have shown that some bioaerosols even carry preferential charge, so that the charging portion of this might actually, which is probably where most of the inactivation would occur, either due to stress from charging or stress from the ozone produced by ionization. But you can potentially eliminate that in some situations with bioaerosols. What we're finding now is that this technology is actually embedded in many bioaerosol collection technologies that are coming out. It has a lot of advantages with little to no pressure drop, because you're not actually pulling through a pore or pulling through a small orifice where you're trying to focus the particle. You don't see a lot of, you don't have to have a lot of pressure drops, so you can do, means you can use like really low energy blowers or fans, which means it can be really quite quiet. Although, which has huge advantages for monitoring in areas like schools or hospitals or other places where a lot of noise would be a big problem. So monitoring for infectious disease in these cases, becomes much more palatable if you have something that isn't so loud. One of the drawbacks is collection efficiency is still an issue for a lot of the existing systems. This is something I think that is an area where I think a lot of work could be done to improve collection efficiency in these systems while still maintaining sort of a quiet and low impact type of collection. And I think that would improve monitoring in a lot of areas. So kind of moving forward, essentially, culture analysis is sort of the, well, I guess, let's see, I'm going to move into analytical techniques at this point. So a lot of the common biological analysis techniques I've mentioned over the course of talking about collection, there's culture, there's nucleic acid based or molecular techniques, which could be either protein or nucleic acid. Then there's also some of the more standard chemical techniques like mass spectra, and then visual techniques like microscopy. And your collection technique and your analysis technique have to be matched appropriately so that you can do them both. Culture, I think, it's probably the oldest way to analyze for bioaerosols. I think the first thing that anyone did was to culture a sample to see what was living in it. And we found lots of bacteria and molds. I think that there's many things now with the advancement of a lot of the molecular techniques for characterizing proteins and nucleic acids, where a lot of the traditional culture methods have kind of gone by the wayside. But I think there's still there's a lot that can be learned by relying on sort of traditional culture techniques like selective media and biochemical tests. You can actually look at the actual phenotypic behavior of the organisms in your system, whereas when you look at it purely from especially when you're collecting something very broad, you can only hypothesize about what, the gene is there, but just because the gene is there doesn't mean it's being expressed or that it's doing it. And so like characterizing that genotype to phenotype transition does require being able to cultivate things, but of course not everything is culturable. So in culture comes with a lot of complications, right? So culture can provide a lot of utility in terms of understanding the phenotype of your organism, what it's actually expressing, what it's actually potentially doing in the system, at least in the cultured system that you have. one thing to remember is that organisms that were cultured from a sample were undeniably viable upon collection, but not all viable organisms can be cultured under known laboratory methods. And this is a big trade space. There was an article back in 2005. I don't think this is fundamentally very different today, but it suggested that only 1 to 2% of all environmental bacteria are culturable and by our current methods. So that means if you're looking only at culture, you're probably missing the largest fraction of microorganisms in your sample. But if you're not looking at culture, then you're also missing something about what can be understood about your sample. So while the numbers are small, I still think there's things to be learned. So this sort of leads heavily into the viable viral aerosol characters cultivation. If you've paid attention to the debate over airborne transmission over the last 2 years, one of the big takeaways is that cultivation of live or culturable viral aerosols from ambient collections is extraordinarily difficult, even, I said in here, even if you know exactly what you're looking for and exactly how to do it. Viruses are all extremely host specific, even specific down to the cell lines. What you'll notice with most of the SARS-CoV-2 work along this lines is that it's cultured in Vero E6 cells or some kind of Vero cells, which is a monkey kidney cell, not a human respiratory cell. These Vero cells are convenient because they express a lot of things, but they aren't the virus's natural host, and that comes with some complications. A broad spectrum live viral assay would be really extremely challenging, if not impossible. I think in many cases, something like a Vero cell is the closest thing we have, because you can actually culture quite a few human viruses in Vero cells, but it's certainly not far from complete. Let's see, if there are lots of examples now, both in influenza and SARS-CoV-2 in particular, of people cultivating virus from bioaerosol collections. I think Bill Lindsley did it in 2010 with influenza. We and Lebnicki did it for SARS-CoV-2 in 2020 and 2021. So besides just the traditional methods, you can also use sort of molecular methods as a well, I would almost say using it as a crutch, although I've actually become much more attached to having the confirmation by PCR showing that even if I'm seeing cytopathic effect or cell death in my system, I also see that the virus I'm interested in cultivating is replicating at the same time. Because if you look at PCR before and after cultivation, and you see that it's increasing, then it does give you the confidence that, yeah, it was the virus that I care about. It was causing the death in the cells or the plaques in the cells or whatever. And so while I think we initially used it as a crutch because cytopathic effect was its concentrations, despite what everyone thinks of viral aerosols in infectious disease situations, concentrations are generally really low. And that makes cultivation really hard because of all the losses in processing. So this comes out in trying to get more and more sensitive techniques and little tricks to try and identify the presence of replicating virus, even if you don't see broad cell death in your system, and without passaging 100 times to try and bring it up where you might make a mistake. So just these are sort of some of the viral analysis methods that we talked about. qPCR, RT-PCR, those are, I guess that would be qRT-PCR if it's quantitative for RNA. So PCR refers to amplification of DNA, RT-PCR, reverse transcriptase DNA, PCR refers to converting RNA to DNA and then doing PCR on it. Western blot protein analysis, these are common. So like immunofluorescence microscopy, I've got a couple examples from our own work. Electron microscopy, again, I think everyone likes to see the thing they're looking for. And I think that's why microscopy is so pervasive even now, even though it's not ultra specific. And even when you show people pictures of a virus, they will complain that it doesn't show enough features of the virus. So, such is life. Serial culture, again, this is one that is used a lot in the cultivation of new viruses, but we've also brought it into bioaerosol analysis. I think TCID50s and plaque assays are like these are the things that what we all want to see. We all want to see that there's an infection, that the cells die, and that it's that virus. And plaque assay is probably the thing that most of us are after the most, because it's the thing that's the easiest to, it's one, I see cell death. Two, I can count the exact numbers of viruses I think are in the system, because they make little holes in the cells. So it's I think the thing that people like the most. Microscopy, as I said, you get a visual image of the collected particle. There's little or no biological information involved here. So if I asked you, what is this picture of, you would say, I don't know, it's some fuzzy stuff and some dark stuff. But I know, because I put it on the slide, that it's actually a collection of spores and some other crap around them. So it's an aggregate of bacillus spores, as it turns out. So, but you couldn't know that simply by looking at the picture. That's part of my point. You have to be really careful how you collect things, that you don't distort the particle upon collection. Otherwise, it won't look like what it did, and you might be fooled about what it was doing in the atmosphere. Nucleic acid analysis, I've brought this up, I don't know, a bunch of times already, because it's super common. Basically, we use nucleic acids to identify microorganisms or to characterize them, to understand their functions. When we go down to PCR-based analysis, we're basically looking for some ultra-specific piece of the DNA or RNA that identifies that organism from every other organism on the planet, or we hope, if it doesn't cross-react. So again, you have to know what you're looking for in order to use PCR. It's very, very sensitive down to the single organism level. If in the actual reaction, that doesn't always translate to single organism in your collection, because you have to process that collected sample and all the dilution steps that go into that. Multiplex reactions can be used to detect multiple different things at the same time. So that brings me to 16S. So this is just a massively, initially the first 16S assays were not, they were basically massively paralleled PCR reactions. Now it's so cheap to sequence things that why bother with some sort of massively paralleled system? We can just throw everything into a tube and sequence every single thing that's in there. And practically speaking, with a lot of aerosol samples, what you'll find is that sample is so dilute that you have problems getting good coverage and good identification of things. And so a lot of times we focus on things like the 16S gene of bacteria, amplify that in the sample. So amplify all the 16S genes in the sample and then sequence that. And that gives you a good idea of what all of the bacterial species were in that sample. You can go shotgun sequencing, which is sort of a step back. So I just want to see everything that's in my sample. You don't need any a priori information. And it's theoretically possible to identify every living thing in a sample by doing this, right? You just sequence everything, see what it is. Fantastic. What you'll find is that you don't know what most of it is. Most of it is just unintelligible sequence information that doesn't map to anything, and that you still have some only some small fraction of it that's ever been identified before. And bioinformatics becomes a problem here, especially when you're talking about environmental samples. And low coverage, as I said before, the reason we do amplification for things like 16S is because there's not a lot of bacteria or viruses or anything in any air sample you're going to collect over any reasonable amount of time. So you wind up with low coverage and then low ability to identify. How am I doing on time, Alex? Am I doing okay?
I think you're still doing good.
Okay, that's good. I always worry about that. I don't have a clock, so. So protein analysis is another way. I think proteins are, it's translated nucleic acid. So this is the functional part of the organism. There's a lot of value, I think, in characterizing the proteins in an organism. If you look at things like we normally do for identification, things like immunoassay are very specific to a specific organism. You can do bulk protein analysis, which just looks at the total amount. You can use this to sort of get an idea of how much bioaerosol, like the bioaerosol mass, just by bulk protein analysis. Biological mass fraction is what that used to be called. But those aren't very specific. You can do some more complicated protein assays to try and look at all the proteins in a system, particularly if you're looking at a specific organism or for a specific organism. Characterizing the proteins can let you know a little more about what it's actually doing. At least tells you what genes it has that it transcribed to use, vice the ones that it might have just not transcribed in the protein. So the protein analysis is a good step towards understanding what an organism is versus what it does. MALDI mass spec is another way to get at protein analysis. I think these are the more, MALDI might be an older terminology, but it's one that I still use. It's matrix-assisted laser desorption ionization mass spectrometry. The advantage of this is that you can actually ionize entire proteins, and so you get very large masses. It doesn't work with every kind of protein, although some of the more modern instruments are getting better about doing more and more kinds of proteins. So, and you can use MSMS to sort of fragment proteins to look at. If you know the protein, you can fragment it. You can look at the protein fragments. You can look at changes on specific proteins, which is a lot of what we've done initially to characterize the impacts of the environment on bioaerosols, is to look at the impacts directly on the proteins. MALDI-MS was actually one of the first things that we did there. Mass spectra, so there's talked about MALDI already, but elemental spectra, things like the BAMs and the SPAMs. I think this is now, the SPAMs is now a commercial instrument made by Livermore Instruments, still Dave Ferguson. And you get elemental spectra for single particles. And you can use some of those elemental spectra to walk back to the potential for something having biological origin, just by the mix of carbons and hydrogens and other things in there. And you can sometimes get an idea of what kind of microorganism it is by the presence of other elements. So since it's a single particle technique, you have a lot of advantages in terms of getting elemental spectra from single particles and being able to map those back to something specific, rather than if it were an aggregate, it would be hard to make that same distinction. This is just a little table in a book chapter I wrote a while back about selecting samplers for downstream analysis. So I'll pass back to Alex for real time.
Okay, great. So just hang on that outline slide for just a second. So thank you, Josh. That was a great overview of techniques. And as he even mentioned going through there, it amplifies one of the things I said at the beginning, which is this is a really multidisciplinary thought process of bioaerosol sampling and analysis. And you can see that even by the choice of the sampler and the analysis you use, there's a lot of questions to think through here. And so it can be complex. Biology it can be a complex thing to sample and keep alive and think about. And so there's no one technique that answers everything you want, as he emphasized. And depending on what you're looking for, there can be trade-offs and things you want to emphasize. One thing that may be important to that is the ability to look at things in real time and get rapid information on a time basis. And so there are things that you can trade away, but maybe emphasize to use some instrumental techniques to look at bioaerosols. So I'm going to focus on one in particular, the fluorescence aspect of that. But before we do that, I'm going to say a couple things. Okay, go ahead, Josh. Josh, can you move the slide? So when we are talking about real time, so one of the things we want to talk about is real time techniques. And there are a variety of these. And first I'll say some things on why you might care about real time. Hey, Josh, are you still able to click the slide?
Sorry, Alex, I seem to be stuck. There we go.
Okay.
Sorry about that.
That's all right. Just wasn't sure if you were taking a bathroom break or.
I got up for a second and came back, but yeah.
Will it not? Oh, there we go. Okay. So yeah, so why do we care about real-time analysis, whether it be a PBAP as I've defined here, or more broadly bioaerosols or viruses or whatever? And so one is that you can do the analysis paired with the collection. And so when we talk about real time, we sometimes use the word online, and these are not really equivalent to each other, but just it sometimes happens that way in people's lingo. The idea is that you're collecting the aerosol, and then the analysis is being done automatically by some computer system and what's going on. And so the benefit of doing real-time analysis is that you can do this continuously. Oftentimes it can be even unattended. You can put an instrument in a remote place, and it doesn't usually require consumables or physical analysis that needs to take place. And so this mimics the same process of real-time aerosol analysis that is in many other areas of the aerosol world, and that you'll hear about in talks and the rest of the AAAR conference if you attend the rest of next week. And so in the lower left, aerosol chemistry through aerosol mass spectrometry, and you can get fine detail in terms of time and all the different properties you're interested in. And so one of the things that we care most about is getting rapid information so that we can then potentially move the collector as a function of time. And you can see in the middle lower, you see that this AMS was put on an airplane and flown around, and you can get spatial information because you get information rapidly on what's going on. In the upper right, it's a complicated slide, but just to show that on the x-axis, it shows time. Each little tick is a day. And so on the lower part where it looks like flames, you have a y-axis that shows the diameter, and the color of the flame shows the concentration of some, in this case, a fluorescent particle. And so you can see real more process level information of things developing over time and size distribution changing that you would never be able to see if you didn't have that time resolution. And so there's a lot that can be gained there. But there's a lot that's thrown away as well, and we'll talk about that in just a little bit. Okay, next slide, please. So I'll talk about fluorescence analysis quite a bit in several slides, but before I get to that, I want to say just an overview of some of the real-time techniques that people use for bioaerosol analysis. And so this is a table that we put together in that same special issue in AAA, AS&T a few years ago. And so we broke it down into these 7 categories, and then the table lists some of the properties of this, but then obviously the paper goes into more detail, overviewing the different categories and giving lots of references. So if you're interested in real-time techniques, this is one of several places that you can look. So fluorescence is something I'll zoom in on in the next few slides, but some of those principles apply broadly to all real-time techniques, and really even outside of the real-time techniques. But elastic and other elastic scattering and other types of photography, Raman spectroscopy, mass spectrometry, as Josh has already actually introduced a little bit, the use of single particle mass spectrometry to look at spectrometry, or to look at biological aerosols. Breakdown spectroscopy, that's where you hit the biological aerosol with such energy, whether it be a laser pulse or whether it be other ways that you can spark induced breakdown spectroscopy, and you basically break it into elements, and you induce and you infer that it's biological based on the elemental composition. There are remote sensing versions of real-time biological aerosol as well, where you shine a light, a laser over kilometers and then collect that information, and you can see an integrated signal over this, or integrated or spatially resolved signal. And also microfluidics, where we collect the information, and then once it's in the fluid, we can send it to various other real-time techniques. And so this is where the whole power of chemical and physical analysis come into play. So there's a whole spectrum of real-time techniques, but this is kind of the overview of them, and we'll zoom in. Okay, next. So to say a few words as for introduction on laser-induced fluorescence spectroscopy, the idea of using laser-induced fluorescence, or LIF, is based in work that was funded by military organizations in the US and otherwise to detect agents of biological warfare, BWA. And there's a lot of work in the mid-90s and 2000s and still into the present. And so this, from kind of an editorial perspective, from my thought, is one of the places where you see the multidisciplinarity of the communities that work on this, because there's a huge amount of information that is wrapped up into these topics that are not easily transferable into the public domain or the academic research domain, because there are certain both proprietary and national security level knowledge that's wrapped up into this. And so in some cases, the communities think and care about different things and don't always flow back and forth as efficiently as you might want. So this is just one of the aspects of working in this field. And so looking at real-time bioaerosol analysis, but especially LIF, the goal from an atmospheric bioaerosol perspective is often looking at the environmental processes. When do certain bacteria or spores or pollen come out, and why? And what effects does that have on the atmosphere or the environment? From a fungal or mold spore perspective, certainly there are human health considerations that you might want to know. Where and when and why are these being emitted? Pollen for human allergies perspective, also bacteria and viruses as well for infectivity perspectives that we care about these things. Okay, next. So from an LIF perspective, again, the fluorescence side, there are a number of instruments, and I'm not going to endorse any one specifically. I will, however, show some data and that is in a bunch of papers that you'll see here. I'm not meaning to endorse a specific or individual products, but there's a lot of work that's been done on several of these. So these are some of the instruments that are available. There's a next slide as well. So the UV-APS is no longer sold, but there's still a lot of data out there that people have shared. The University of Hertfordshire in the UK developed a WIBS that was eventually licensed to Droplet Measurement Technologies, or I guess they're now called Droplet, in Longmont, Colorado. And so there are differences I'm not going to get into here on this slide, but they use different wavelengths of excitation. That'll make a little bit more sense in a second. But more or less, they use the same process of shining light on something, measuring the fluorescence that comes out. Okay, so next slide, please. And then here are some others as well that show the same idea: the BioScout, the BioVigilant, using a 405 nanometer laser. There is the Rapid E, which and also the DMT SIBs. Actually, I'm not sure they're still selling the SIBs, but same idea, but you get a lot more resolved information from a spectral perspective. University of Hertfordshire made something called the MBS. So this is kind of a beefed up category of what we just talked about before. And then there are a variety of others that are only using scattering and not fluorescence. And honestly, this slide is not quite up to date. There are some others and some smaller and cheaper ones that are now hitting the market as well, especially focused on pollen analysis. That use the same general principle, shining light, and you get fluorescence, or you get a pattern of scattering and holography that you can try to figure out what all of this information is telling you, to both detect if it's fluorescent, to detect if it's biological or not, if it's a pollen or bacteria or spore, and then in a lot of cases trying to guess at what the species or the genus of that particle is. Okay, next. So I have a lot of information I could share on this. I've collapsed it into a few slides, but even that I'm going to do relatively rapidly because I don't want to hammer you with the physics and the chemistry too hard here. I do want to say here that the concept of fluorescence is that you shine a light of one wavelength, it is absorbed. Some of that, what we call vibrationally relaxes in the form of heat, and then some light comes out at a little bit lower wavelengths, a little bit less, a little bit longer wavelength, which is a little bit lower energy. And so there are only certain things that go through this molecular process of fluorescence. And so it can be somewhat selective for classes of molecules. What's important to know about fluorescence spectra is that they're really broad in nature, whereas other spectra, infrared spectroscopy, mass spectroscopy, can be really narrow and highly resolved. But fluorescence, it's not because of the detector, it's because the physics of what's going on inside the molecule are always going to be broad. And so they're not, it's not possible to look at the fluorescence spectrum of a complex system like an aerosol and get really refined molecular information that helps you get at exactly what something is. It's always relatively uncertain, and it groups things more broadly as you can ever get by PCR and some of the things that molecular techniques that Josh was talking about. Okay, next. But there are a class of compounds that fluoresce. Anything that has what we call a substituted aromatic ring, depending on whether you're a chemist or not, this is either really obvious or you may have never seen this before, but these ring-like structures here, these hexagons, kind of at the top of each of these molecules, if you have certain substitutions there by adding extra stuff to it, these are the general features that exist in molecules that fluoresce. And so there are a lot of different compounds, aromatic amino acids, coenzymes and certain flavins, NADH and riboflavin, pigments like chlorophyll, structural polymers like cellulose are in the categories of things that fluoresce. And so I won't give an overview of that. If you are interested, go to Christopher Pilker's paper from 2012, where there is a 5-page table on the molecular information of biological fluorescence. So that's where you can get a lot of information there. Okay, next. And so it wraps to this, which is, these are called excitation emission matrices, where on the y-axis, it is the wavelength of light that we are shining at the molecule. That's the excitation. And then the emission is on the x-axis. That's what comes out. And so if you pick a wavelength at any one horizontal line, this is what you would then shine at the molecule. But we want to shine a light that hits one of these dollops of light here. In the upper right, you see 6 different compounds that show the region of fluorescence that it is possible. So if you shine a light somewhere that is longer in wavelength than something fluoresces, you're not going to see anything. You have to tune your light to make sure that you're shining it on the right place. But based on this, we can be somewhat selective on the types of compounds that we're seeing depending on the fluorescence in certain places. Okay, next slide. And so this is kind of a summary of that, where we have a couple different instruments where so on the same excitation emission matrix on the y-axis, we have the excitation, and you see a bunch of lines at about the 350 nanometer mark, and then there's a bunch that are scattered kind of in the 260 to 280 range. And this was done early on to focus on 2 different regions. The 260 to 290 region is used approximately for protein fluorescence. The idea is that aromatic amino acids in this region will fluoresce no matter what's going on with the molecule or what's going on with the biological particle, I should say. And then this region around 350, you pick up NADH and NADPH and riboflavin. Originally, it was meant to pick up cells that are metabolizing, because these are cell metabolites that go away after the cell is no longer metabolizing. It's not nearly as simple as that. It's much more complicated, but by putting together a sequence of excitation wavelengths, you can start to differentiate between different cell types and different biological particles. And so not all instruments are the same. That's important to know. But there are some broad differences, broad overlaps. And I said this is a bit controversial. I actually didn't take that off of a slide that I presented several years ago. I don't think that's controversial anymore. That if you see fluorescence at 350, it used to be assumed that there was some viability to that particle, but that's a gross oversimplification of things because there's a lot going on, some of which I'll say in a second. But just to say, it's important if you compare the data from one instrument to another that you don't think laser-induced fluorescence is the same everywhere. There's a lot of technical differences that really come into play and are important. Okay, next. So like I said, one of the purposes of using some kind of a real-time technique is to be able to get two things among others. One is time-resolved particle information, especially time-resolved particle size, which is great. And so on the upper left, it just shows an example where you can see the particle size in the right parts of the size distributions, the right parts of those upper left quadrant. You see on the x-axis, you have particle size. And so there's two size distributions, one which have on the bottom, which has a double hump, and the one at the top that has a single hump. And these are portions of just narrow periods of time. The one at the bottom is half an hour or so, and the one at the top is a couple hours. But you can use these instruments to differentiate behavior in particle size in relatively narrow windows, which is really useful. And because certain particle types have characteristic sizes, and so you can start to understand what it is, but you would never be able to do that if you didn't have the ability to look in real time. And so like a lot of particle instruments, you have to pay attention whether it's aerodynamic size or what type of particle size. I'm not going to get into that right now, even though I put it on the slide. Most of these use something called optical particle size, where you make an inference of what the physical size is based on the amount of optical scattering that comes out. Some of these instruments also provide something close to a shape factor. It's not usually rigorously defined in the same way it is in some places, but there's some you can learn a little bit by the asymmetry of how the light scatters about approximately whether it's spherical or not. And I think that's all I'm going to say here. So move on to the next slide, please. The instrument response, this is where I'm going to narrow in on one specific instrument, but the principles are still broadly applicable to other, especially laser-induced fluorescence instruments, where if we have a couple different wavelengths of excitation, so look in the lower left for the moment. So notice, remember I said that there were these two regions of excitation. If you draw horizontal lines across something in the 280-ish region and the 350-ish region, but then the detector, sometimes you may measure the full fluorescence spectrum, highly resolved, but other times you may just group the emission into a block. And the WIBS, for example, has two excitation wavelengths. So there's two lights that shine on it, and those would be represented by two horizontal lines here. But we only collect it into three total emission bands. So we're just these three chunks. And so basically we have particle size and then three parts of information for every particle that we get. And so does it fluoresce in the A channel, B channel, and C channel? We have independent answers for these. And so based on those three, there's this little Venn diagram in the upper left, whether it's yes in channel A and no in channel B or what. So it can be an A, B, an AB, ABC, whatever. The combinations of these three fluorescent emission channels that we see already gives us a pretty complex set of possibilities, even in just these 7 plus non-fluorescent. And so in the upper panel, you can see size distributions of certain pollen, dusts, fungal spores, bacteria that were analyzed. And you can see the color represented here matches the color shown in this little Venn diagram on the left, where you can see the flavor of fluorescence basically looks different, and the distribution of those looks different based on what material we have. And so different fungal spores in the middle have a different breakdown of these fluorescent types compared to another fungal species, whereas the bacteria look relatively similar. And so you can see even from this that the type of information that we're getting is wildly less than what we get out of the molecular techniques and some of the other, and culturability and all these kind of things that we talked about before. But we have this information in real time, and so that has a benefit to it. And oftentimes we use real-time techniques in the outdoor environment or in a sampling environment indoors to, let's just say, the WIBS or the UV APS or even mass spectrometry or something, and you get an idea of what's going on and when things happen or if something is happening. And then if you really want to dig in and see exactly what the species is, then you have to apply other techniques. But those techniques maybe are too expensive to do all the time, or maybe you want to run them for 2 hours to really hit the place that you're the most sensitive to what's going on. So you can use a real-time technique as a survey of sorts and then zoom in with other techniques. Let's go ahead and go to the next slide. Two important pieces here, false positives and false negatives. False positives just means you think you see something that you think is biological in nature, but is not. And so in this case, it shows the flavor of fluorescence associated with various things that could also fluoresce. Soot in the outdoor environment especially can really cause a problem, as it looks a lot like a bacteria, sometimes in particle size, but also in fluorescence. And in an indoor environment especially, bleached cotton fibers, just fibers of all kinds, can really cause a problem. And so you have to be really careful in an indoor environment, because cotton t-shirts, people wearing clothes, bed sheets in a hospital environment can really cause problems. So this is where you can use the laser-induced fluorescence as a survey, but don't use it as the end-all answer of biological particles. Okay, next. So this is false positives. This is false negatives. There are also things that you want to fluoresce that don't fluoresce. And so certain biological particles that have either are small cells and don't fluoresce strongly, or are just small and the detection limit is hard to get at, or they have poorly absorbing cells, or they have strongly absorbing cell walls. If we have certain kinds of fungal spores that have dark walls that don't fluoresce real well, cladosporium is hard to detect by fluorescence in certain applications. And so you may miss some of the things you care to see. Let me do, Josh, we're going to do 2 more slides quickly here, and then it's going to transfer to you, but I'm going to have you skip for a while over stuff, and I'll say that in just a second. So I'm going to give it back to you after 2 more slides. So the most basic question and the most basic way people think about fluorescence analysis is just, is it fluorescent or not in a certain channel? And so that has been a fine way to do this, but it's a relatively coarse binary perspectives. But even the definition of fluorescent or not implies that you have a threshold between the two. And so there are various different strategies that to apply to decide whether it's fluorescent or not. And so if you get into this game, I would suggest that you think about these questions and read some of the literature, getting at the answer of how do we know if it's fluorescent or not, and what does that even mean, and how does it play out? So there's one reference at the bottom that you can look at by Savage et al. And there are others. Okay, next slide. But then ultimately, if you really care about this, using fluorescent spectroscopy to look at biological particles, there's a whole other class of analysis techniques, which is clustering. There are machine learning and algorithms that basically dump all the data in. This is going to be a really simplified version of this. If you do machine learning, I apologize, but just for the sake of the 30 seconds here, the idea is that you have a lot of data on many individual particles, as opposed to just a collection on a culture or something like this. We now may have tens or hundreds of thousands of particles, each of which have their own fluorescence and size properties. We can dump that into certain analysis techniques, and then it clusters them into categories that we can then make inferences on. And so there are various techniques, and there are various research groups that really put energy into refining these techniques. So if you get into this kind of analysis, again, look into the literature and see what is available there. So with that, Josh, I think it's best to just skip over my next little section where I was just going to give a really broad, so just go next to your,
Okay, so yeah, and this is just a couple of things, and I'm actually going to make this actually really quick as well. Not going to belabor too much here because I want to leave a little bit of time for questions. Sorry, I'm having trouble flipping slides today. So I'm gonna, a lot of what we've done, I was gonna talk a little bit about what we've done over the last couple of years in terms of sampling in the clinical environment and the other kinds of bioaerosol work that we've done. This is just some examples of how we did some sampling in some of our clinical spaces. A lot of times this comes down to, the decisions you have to make about what you're going to sample and where you're going to sample have a lot to do with the rest of the environment that you're working in. And so a lot of times when I'm trying to learn something or trying to do something very quickly, I grab what I can and what I think will be tolerable. For instance, in this case, in the rooms of people who are sick with a disease they don't understand. And then none of us do, which is why we're doing the work in the first place. And so you have to be a little bit, you have to be cognizant of your situation in many cases. But even still, you can get quite a bit of good data this way, looking at sort of what samples are positive and which ones are not. We found initially in a lot of our work that a lot of the air samples were positive. And so were our air handling grates, which both indicated aerosol in a disease. This is all around SARS-CoV-2, of course, which indicated the likely presence of aerosols containing virus. When we wanted to know more about the size, I had intended to talk here a little bit about the combination of real-time and collected aerosol techniques and how sometimes even all of your best efforts don't lead to much. I really hoped we'd be able to look at the APS data and get an idea of the background, of where the human portion of this aerosol distribution was. In fact, I could not mathematically do that, despite most of my best efforts. But even with the NIOSH, we were able to collect several samples that replicated in cell culture. This was confirmed by serial PCR in this case, and then also again by SEM imaging. We talked a little bit about that already. I did want to talk about this sort of at the end. Other folks have been doing this, so I wanted to mention the VIVAS sampler, which is essentially humidity controlled. You basically grow up the bioaerosol particle with water and then collect it, which should take a lot of the impact out. Lednicki and C.Y. Wu's group at University of Florida has had a lot of good luck collecting very viable virus samples from there. However, I wanted to talk about this from sampling in general. So we had some good luck sampling with gelatin filters. Lednicki and C.Y. Wu had good luck with this Biospot Vivas, which was very, very carefully controlling the aerosol, the forces on the aerosol. However, Ratnasar Shumate et al. Published a paper where they compared a whole lot of different collection techniques, everything from just collecting onto a PTFE filter all the way to doing something like a biosampler and an all glass impinger. And what you can see is that SARS-CoV-2 aerosols, when generated in the laboratory, can be recovered from all of these. The least, maybe this glass midget impinger in this case, and these two midget impingers probably had the least recovery. But even still, most of these had very good recovery, near theoretically predicted. And so, this may indicate, I think, that a lot of the things that we discover in the laboratory that seem to work well don't always translate to the real world field environment. And there are a variety of reasons for this, particularly with regard to viruses. It could have been the cell line that it grew in. It could have been just it's not as we have a little more advantages from in a laboratory environment than we do in an actual human expelled particle. Sometimes the bioaerosol field work that we do relies heavily on real-time measurements and tracer particles. And so this is another combination of real-time equipment, the fluorescence equipment in this case, measuring at 405, and a uniquely biotinylated tagged PSL particle. So we're actually using DNA-based detection to identify our tracer particles, as well as real-time detection to identify tracer particles. If you want to see more about this, go to watch Sean Kinahan's talk. And then, when we want to learn about what happens to specific aerosols in the outdoor environment, we sometimes have to take our lab into the field. this is one example of that. These are giant rotating drums. Here's another one. These are a little smaller, large rotating drums that we took in the field. And it allowed us to look at the impacts of a lot of things like sunlight and other gas phase species directly on in the real environment on not just the viability but also fluorescence. And so coming back to a little bit with what Alex said a minute ago with regard to false negatives, real-time fluorescence in particular is impacted not only by the sort of intrinsic properties of the bioaerosols, but those properties, the structures of the amino acids, the other fluorescent molecules can be changed enough that they no longer fluoresce in the same way once they've been exposed to an outdoor environment for some period of time. And that's a function of what the environment is and what the factors are. And these are the kinds of things that we can learn by doing these sort of controlled releases in ambient environments where we both control and contain the particles that we have. And I'll probably end it there so that we have, 3 minutes for questions, but I'm fine staying around a little longer if they don't shut the Zoom off.